Hi, this is Dr. Jabali, and in this video, we're doing topic 2.4, which is all about proteins. We're going to focus on previous IB exam questions, which have questions related to the topic 2.4, which is about proteins. So we're going to look at the written answers. These are usually the essays and the short answers. Now, the most important thing about IB when it comes to these short answer questions is to always read the first word, which is the command word, what they say, outline, describe, explain, because that tells a lot. The second thing is to always check for the amount of points that it needs, because if it's a large amount of points, that means you have to get as much as detail as well as many broad answers. And if it's just 2 or 3, then that means you have to go very much straight to the point. Now, in this question here, it says the structure of protein. Now, the structure of protein, we know that protein is made of 4 levels. There's the first level, the second level, the third level, and the fourth level. Now, the first level is usually just a chain of amino acids. And of course, these amino acids are attached by a peptide bond. All right? And I know that it's completely linear, right? It's just a simple chain. And we know that there's about 20 amino acids, and we can have different mixtures of these amino acids. Then we have the secondary. The secondary is basically where they start to bend. Like this case here, that's a beta sheet. And then we can have the spiral like this, which is the alpha. Now, the beta sheet and the alpha, these two are secondary structures. And the reason why they bend like this is because of their hydrogen bond. Now, that's more of the bonds that is happening. Usually they're happening around here, these bonds between amino acids. So the next thing is that we have is the tertiary, where the tertiary in this case, this would not only be both the peptide bond and the hydrogen bond, but it would be a mixture of them. So in the tertiary, you probably would have the zigzag and then the spiral, then the zigzag, then a spiral. And that means your combination of 1 and 2. So 1 plus 2 is coming together. That means you do have peptide bonds, you do have hydrogen bonds, but in addition to that, you add disulfide bond. That means the R groups are basically binding now. They bind with other R groups. So the structure is more like an example that would be the enzyme. And then you have the quaternary, which that means there is 2 or more polypeptide chains. That means it's not just one chain like the first one here. You have 2 or more. And you also, in this case, you have conjugated molecules. In this case, it can be vitamins, it can be minerals, such as calcium, iron added to it. And in this case, you also can be adding with all of these. That means you have as a quaternary, you have peptide bonds, you have hydrogen bonds, you have the alphas and the betas in there. You also have disulfide bonds, which they are group attached, and with that, you have the conjugated molecules such as calcium or iron. And an example of that would have been hemoglobin. Now, I know that this is more like an HL content, but I have seen sometimes fall on SL by mistake. So I always make sure my SL students need to know the structures 1 through 4 of a protein. So let's look at the mark scheme. The first thing we have is the primary structure, as we have mentioned. And as you can see it here, it is a chain of amino acids. The second thing, the position of why there's 20 different amino acids. Third is linked by a peptide bond, as we mentioned. The secondary structures, that means you have the amino and the carboxyl groups happening. Now, that's basically the end of one amino acid to the other. So, for example, here we have a hydrogen bound to a nitrogen right here, carbon bound to a hydrogen, plus an R bound to this, and the OH. If you want to combine this amino acid to another one, usually here you will have another amino acid come in, and what would happen is that right here, the OH and the H will leave, and there would have been a connection between the hydrogen and the carbon of the other group, and this right here would have been the peptide bond. So basically, it's between the NH and the CO here. We're talking about these 2 areas, this area and this area. This is where the connection will happen between 2 amino acids. We also have hydrogen bonds formed. Now, what it means by this hydrogen forms, I told you if This is when it's a secondary. They're starting to fold like this, the chain. And these foldings are happening because there's weak hydrogen bonds connecting between the amino acids right here. Moving on. So basically we have the alpha helix formed, which the peptide calls up, is basically the joint that I was doing like this. We also have the beta sheet form, which is the zigzag that I was talking about. So this section here is a secondary, this is the primary. And then we have the tertiary, which is folding up polypeptides. Of course, in this case, it's just folding, like, for example, I showed the drawing of alpha and then the beta and then the alpha. It's just adding up the together. And this has disulfide bridges. It can have a hydrogen, ionic, or hydrophobic bond. These are 4 different types of bonds. And these usually happens with the R groups. So far we have mentioned here the primary, we have mentioned the secondary, we have mentioned the tertiary, and now we got to the quaternary. And this is many polypeptide chains, as I've mentioned. And you have conjugated proteins, as well as non-protein molecules such as calcium and lipids and stuff like that. So the examples will be here: metals, nucleic acids, carbs, lipids. These are examples of it. So when we talk about the structural protein, they're talking about the 4 levels. The 4 levels we have here is 1, and we got the second one, and then we got the third one, and then we got the 4th one. So as long as you know the traits for each one of those 4, you are completely fine. So once, just as a recap again, make sure the first one is just about this chain of amino acid with peptide bonds, and there's about 20 different of them. Secondary structure is all about the hydrogen bonds and forming alpha and beta. The tertiary one, it's about frigid hydrogen ionic bonds, so there's a lot of folding going on. Quaternary is more of many polypeptides with conjugated proteins, examples, metals, nucleic acids, carbs, and lipids. So make sure you know those 4 levels. That will be describing the proteins. So let's go to the next question. So the next question here, it says describe the significance. That means significant means the importance of polar and non-polar amino acid. So we're talking about what is it about each one of them. So let's look at basically what is the importance of polar and non-polar. Now when it comes to that, the first thing you do is always tell what they are. So for example, polar in this case, polar, it has charge. And if it is charged, it means that it's hydrophilic. That means it can easily mix with water. That's for sure. And if it can mix with water, that means it can create hydrogen bonds, right? Because remember, a water can connect with other molecules, anything here, through the hydrogen bonds. Now, not only is this good, because for example, let's say you have a cell membrane here and you create a channel that is a protein channel, right? So the things are coming in. This area of the protein has to be polar because it has to be in contact with what? With the water or liquids that it's in here. So part of the protein needs to be polar. So for example, it's very important that a part of the amino acid is polar in order to connect with the fluids. And then on the other hand, we have non-polar section of the protein, and that means there is no charge. And if there's no charge, it means that it's hydrophobic. And if it's hydrophobic, it doesn't mean that it mixes with water. So in this case, they usually will have hydrophobic interactions. They don't touch with anything. So where do they usually found? They are found basically on the interior of the protein. And what I mean by the interior, that means this area here. Let me highlight the interior side of the protein. That means they're not exposed to the water or the liquid that is coming through the channel. They usually are in the area here where it is connected with the fatty acids of the phospholipid layer. So they are really touching the fatty acids of the phospholipid and not wanting to mixture with the fluids of the cytoplasm or with the fluids of the outside of the cell. So usually they are not much mixing with things, but they're more mixing with other proteins. So these are the difference between the polar and non-polar when it comes to amino acids. Now let's look at the mark schemes. So for the mark scheme, as you can see, for maximal mark, response must have polar and non-polar amino acids. So that means we have to talk about both. So we have possible 3 marks for each. So first is hydrophilic for polar and hydrophobic for non-polar. Second, this polar, because hydrophobic, because hydrophilic, because it mixes water, it has a hydrogen bond with water. But the non-polar, it does not have any mixture of water, so it forms van der Waals, or you can say hydrophobic interactions. With who? With other hydrophobic amino acids, because hydrophobic mixes with hydrophobic. Next, it's found in hydrophilic channels, so that means it's in the proteins projecting from the membranes. It's found in posterior interior. So this is basically when, like I was mentioned here, you have the cell membrane, and then you have the protein, and then the cell membrane continue. So basically the hydrophilic would have been on this side of the protein channel, while the hydrophobic would have been on this side of the protein channel. So the hydrophobic is away from the exposures of things. So this is found on the surface, and this is found in the interior, as this is basically saying here, this area right here would have been the surface where it's touching, right? Let me put in black. This is where it's touching everything, and this is the interior where it's not touching the outside, which what I mean by the outside is that we're not touching the water on the outside of the cell or the water on the cytoplasm on the inside cell. So that means they're only inside of the fatty acids. So that's pretty much the questions for these two. Make sure you're talking about they're being hydrophilic, hydrophobic, talking about the proteins being on the inside or the outside of the channels, and what kind of bonds they're forming. Moving on to the next question. Here it says list 4 functions of proteins. Give an example for each. So this one, I'm just put the whole thing all together because this is something about memorization. There is no other way. Now, while you go for a 2 year of IB, you eventually will know so many proteins, so this will become a very easy answer to go. But here are some examples. Now, storage. Now, many people do not talk about zeaxanthin or casein in class, which is totally fine. But you can also store proteins. You can also transport. Now, this is more common, especially when you're going to talk about blood. You're going to learn about hemoglobin. When it comes to hormone, you're going to learn this in topic 6.6, which is talk about insulin, and eventually you will have TSH, FSH, and LH. These are very common for 6.6 on reproduction system. Some of you guys will learn about growth hormone if you do HL. Receptors. We have receptors in the nerve system, and all these receptors that receive, such as neurotransmitters, they are all proteins. So all the receptors in the cell membrane are neurotransmitters. Movement. If you're an HL student, you're going to learn about actin and myosin, which is the muscles contractions, and these muscles are made of protein known as actin and myosin. Defense. If you do the immune system, which all of you do, you're going to learn about antibodies and hemoglobins. These are 2 examples of the immune system that fight against these bacteria. And got enzymes 2.5 or 8.1. We have catalase and ribonuclease. Don't worry about these enzymes. These are more for the HL students, but we know that there is enzymes in our bodies that are breaking down. Structure, we have collagen, keratin, tubulin, fibrin. These are all different types of structures that we found around the bones in our body for structure, for keeping us standing. Electron carriers, this is what you're going to find in the photosynthesis as well as cell respiration. You have pigments. Now, opsin is example of pigment. Another pigment can be chlorophyll. Active transport, in this case, is sodium potassium pumps. This is something that you need to know from 1.4. So basically it needs ATP energy to move things around. We also have facilitated diffusion, that's a channel. And there is aquaporins, which is in the kidneys, which if you're HL student, you will know. So the reason why I put this up here is just do not panic. Make sure you know at least 3 to 4 examples of proteins. Now, I'm going to go over again this and just highlight the ones that are SL. The rest, the HL, eventually will learn with time. So things that you can know from HL easily, it's transport hemoglobin. You have hemoglobin insulin, as well as these ones eventually you will know. You can talk about receptors like the neurotransmitter receptors. We can talk about the defense system. Enzymes, it doesn't have to be these enzymes, but you can talk about the enzymes of pepsin, which you will know later in the future. You can talk about structures such as collagen, the electron carriers. You can talk about active transport and facilitated. So there's many examples SLs can know for the future test. Moving on. It says here, outline the process of protein denaturation. So first we have to know what denature means. Denature means is basically the unfolding of the amino acid chain. So usually what happens is that the structure, how it is folded, this chain here, it will give the function of that specific protein, right? But if that thing goes to something like temperature, a very high temperature, or they put in a pH that it's not its own pH, what happens is the folding will end up losing its structure, and then if you lose its structure, it cannot function anymore. And that's what denature means. It's just that they're losing their way of functioning just because of structure. Now, what will do? The temperature will cause this unfolding to happen. And that means very high temperature. And pH, what it do? It would changes the charges of the amino acids inside of it. So that's what denature means. It's just unfolding a protein, and eventually it cannot have its own function. Now, when we say the change here, the unfolding, there's another word that we can use, which means is change the conformation. Now, what does the conformation mean? The word conformation is the same thing as saying it's changing its shape. So changing the shape of the protein structure can also change its structure. And when you're changing that shape, right, that means what you're doing, you're breaking all the possible bonds that are here. You're breaking so many of the bonds that keeps them folded. So you're breaking the connections between them. So these are all things that you can mention about protein. So what could you mention? There's the unfolding of the amino acid chains, which that means it's changing its conformation with the shape. Now, who can change that? Very high temperature as well as pHs. What do? They break all the bonds that are happening inside of the proteins by breaking these bonds, and thus eventually lose its function. So these are things that you can mention. Now, another thing you can also mention that I mentioned here is that it can also change who. They can change the active site. Now, what is the active site? That's the entrance of an enzyme, for instance, and it can change its shape. So, for instance, let me put here another drawing. Let's say the enzyme had a shape like this, and then because of temperature, high temperature pHs, now what happens to the active site? Now the active site looks like this. So you can see that there has to be a change in the shape of the active site. And by changing the shape of the active site, that means the substrate that looked like a square cannot go in anymore. So if it's a square, it's stuck. So that means the enzyme stopped functioning. So let's look at the Marxium and see what they have. The first thing says, oh, there you go, change conformation, using that word, or shape. It can be the charge structure or 3D shape. So anything change in shape is good enough. Next, the bonds within the proteins are broken or changed. Like I said, the bonds are gone. Third, pH and temperature. That means cause denaturate. So that means you have to tell who are causing the denature. The persons who are actually causing these denatures is the pH and the temperature. Vibration and heat of high temperature. Yes, so this is basically explaining how basically high temperature, right? Because we mentioned temperature can do it, but how? By having high temperature that can break the bonds. Next, high pH or low pH, extreme pH can alter the charges. Just like I mentioned, the amino acids, they have charges, some of them plus or negative, right? Positive or negative. But with the pHs, they can change those charges. That means it can go from plus to negative, negative to plus, and that can change the entire structure. And that can also cause the breaking bonds of ionic bonds because they are plus or minus. Now, proteins cannot carry out its function. That's like I said. Or, for example, the active sites cannot bind substrate. So this is exactly the things that I was telling you. It all starts, so overall, starts by changing the conformation. Next, it's breaking bonds. Next, mention there's the pH and the temperature. Explain how these two. First, the temperature is high, has to do with high temperature. pHs, you can talk about being high or low. What does it do? It changes the charges of the amino acids, so breaking the ionic bonds. And then when all of that causes break, right, change, what happens? It cannot function, or the active site, for example, cannot bind the substrates. So this is a combined, first mentioning what is actually happening, and then what are the two things that causes it to happen, and then giving examples. Now here's the next question. The question says, draw the molecule diagram of amino acid to show its general structure. So this is something that you have to know by heart, and that will be the amino acid here. We have starting with nitrogens and the hydrogens, then you have the carbon and another carbon. This is bound to hydrogen, this is bound to R, this is bound to oxygen. So this is a very generalized structure of an amino acid. But drawing this is not enough. You have to make sure label this as amino group, and you have to mention that this is the carboxyl group. You also have to mention that this here is the variant. This is where you change its structures to make the 20 different types of amino acids. So this will give you more of the points for the IBE. So let's look at the mark scheme. So here you can see the drawing that I had done. You have the nitrogens here, and you have this. Now this is more like a condensed structure. This is basically chemistry, just putting all the elements together. So you can do that or both. Usually I do this one. It's much easier to draw and remember it. So the points here, first you get the COO group at one end, which you can see that this is it. And you have the NH2 on this side. And then you have the CH in the middle with the R group, which is these guys right here. Now, if you show the expand, the carbon oxygen must attach to the C, as it does there. And you show non-expand, the N amino group must attach to the C. Now, what is basically this trying to say? So here, for example, you see the bond is attached to O. Do not do this H and then O, because that is wrong. And the same thing here on is attached to C. So you cannot say NH2 and attach. You can't. It has to be the nitrogen that's attached to the carbon. And here it is the oxygen that is attached to the carbon. So you have to be very careful. That's why I prefer doing the opening wing, so I don't make the mistake of, instead of putting NH2C, I actually have it open and I know the N is the one that's bound to the carbon right here. So open up, it's much easier to do the drawing. Next question here. It says, draw the molecular diagram to show the condensation reaction of two amino acids to form a dipeptide. Here it says the same thing, draw annotated diagram to show the peptide bond that is formed. Now, if you look at this, it's two different questions, but they're both asking exactly the same thing, but with different words. So basically, here it's saying as condensation reaction, here is saying as peptide bond formed. Now, they are talking about the same thing, but in different words. In the end, it's still using two amino acids, which is a dipeptide, and this part here. So where do I see that it's the same thing? The word formed, because condensation is about building something, so you're forming something, and the word formed here is the same. And the peptide bond, because I know the peptide bond is the bond between two amino acids. So basically what they're saying is you have amino acid attached to another amino acid, and this is the peptide bond. Now, a lot of students do this drawing, and you're not going to get points for this. You're not. You might get for this maybe the peptide bond, but you're not going to get for these because this is too generalized. You have to draw it in detail. So when we're drawing in details, this is what it will look like. You're going to have the hydrogen bound to a nitrogen, right? Bound to a carbon that has a hydrogen with the R, bound to a carbon with oxygen OH. Now, you have to show the condensation reaction. So you cannot just go and draw the dipeptide automatically. You have to show it. So you have to put a plus because you're adding that to another amino acid. So you put in here hydrogen, R, and then you have here C with the double bond with OH. Now you have drawn the two amino acids. If you want, you can go ahead and label this as the amino group. And you can label here in the corner the carboxyl group. Sometimes you do get points for doing this label like this. Next, then I would do like a dotted line surrounding these two here. Why? Because it means these two are going to combine. Then I have to put an arrow because it's a chemical reaction. And these two will be the water, right? This is becoming water plus, only put the plus a little bit further away here. Plus, and then you have to connect these guys. So what you're connecting is, if these guys are gone, C was going to attach with the N. So I have to draw again the structure. But this time I'm going to draw them combined and have carbon with the O here. Now here you can see that that's where it's going to combine with the N, with the H, the carbon with the R, with the H, and then the carbon with the O and the OH. Now, is it done? No. There's one more thing that is very important that you put it is show the symbol where the peptide bond is. That's very important. Many students forget to write the peptide bond. Show. The peptide bond will always be between the C and N. So make sure to show that because you get points for showing that. So let's look at the mark scheme. So here the mark scheme, you can see, again, I did the whole drawing, right? Of the two amino acids with the R, the H, and the N's we have here. You also see that I have highlighted the OH with the H, which eventually is the water. Notice that I put the arrow going down. And then notice here that I have shown the peptide bond between the C and N. All of these points can get, because look here, each amino acid group at one end and NaOH on the other, which is true. So you have this on one end and you have this one end. Both are needed. See, that means you need for both of them. It requires a double bond to be shown between C and O. And see, you have to put those double bonds, otherwise you don't get the point. CH in the middle with our groups, which are basically these guys right here. Peptide bond correctly drawn between these two, which is exactly being drawn here down. CO group at one end, dipeptide, and the other one at the end, which we do have that again. See, both are needed, and the loss of water, which is basically here. So this is how you get your points. So make sure you're able to draw this entire thing that I draw without forgetting. Now, I did not need to write these guys, but I do write it because sometimes in mark schemes they ask you to label them to show them who they are. So why not remember their names? Now let's move to the next question. Here in the next question, it talks about the enzyme Rubisco, which is used in carbon fixation, which basically it's used basically in photosynthesis, right? And identify 4 other examples of proteins that illustrate the wide range of functions of this group of biochemical living organisms. What is this asking again? This is asking you again to give examples of other proteins. It's just like that previous question with so many proteins examples. So here is a list again of so many other proteins that eventually throughout the 2 years of IB you will know. Again, one is the insulin receptor, okay? The other one is the leptin, which you will learn in 2.6, which is a hormone. We have hemoglobins and antibodies, which you're going to learn in the immune system. We have keratin, collagen again. These are basically structural proteins found next to the bone. We have microtubules, which you find in the cell, especially in the cytoplasm. We have ATP synthase, which is found in the photosynthesis, as well as cell respiration, which is an enzyme. We have hemoglobin. We also have the sodium-potassium pump. And here it says any other example in function, but look at this, it can be awarded only once. That means there's so many proteins. So here's the ugly part about IB. They will only accept these guys. But if you have an additional protein that is not on the list, they will accept, but it's only once. A second example with a function, so it will accept the second one only once. So that means if you have 2 more proteins here that does not include in any of these guys, they will allow you. So they give a huge list, but then they allow you. So for example, you could say myosin actin for the muscles, you can see pepsin for an enzyme, and you can see that it's not on this list. So that would have considered, but only 2. So if this question had 5 points, that means you could have come up with 2 points out of your own that is not on the list, but then these ones would have to be target. And that's the bad side about IB, because you can come up with 7 proteins and none of them are in here, but you only get 2 points because that's what they're asking here. So that's why I say go get exposed to mark schemes and know which ones are so common to see in the IB mark schemes. And I'm going to highlight here the most common ones used by IB. The most common ones used by IB is insulin. It is the antibodies hemoglobin, the keratin collagen, the ATP synthase, and the hemoglobin. These are sodium potassium pump. These are the most common written in the mark schemes when it comes to proteins. So make sure you have at least these 5s in your head all the time, okay? Let's move to the next question. It says here, describe the secondary structure of protein, which was actually the first question, but this one just wants the secondary. So when we know this, there's alpha, right? Helix and beta sheet. Now, the most important thing is mention the whole word. If you just say alpha alone, you're not going to get the points. So alpha helix and beta sheet. Now, the alpha helix looks like a spiral, while the beta sheet looks like this. They're both are just one chain, but they're folded. Now, what caused the folding? It is the hydrogen bond. So you have to mention that. So these are the 2 structures. What else could you say about this? Nothing much. It's just they're both alpha and helix. There's 2 structures. Now, basically, these are still what proteins, so they're polypeptides, right? And they're folding with the hydrogen bonds that is happening between the groups. And they're basically found everywhere, where that means they're very much like They're found in enzymes, they're found in hormones, protein hormones, because you don't stay a straight line of an amino acid. Obviously, they're going to turn into these foldings. So it's very common to see secondary structure in most of the proteins. So let's go for the mark scheme. So polypeptides wound into helical structure, which is the alpha helix, and we also have the pleated sheet, which is the beta one. Second, stabilize, held in shape by due to hydrogen bonds. So you just say it has hydrogen bonds, what makes their folding. And it's regular and unvarying with things. So that means they're found everywhere. See, a lot of annotated diagrams. The annotated diagrams was what I just put it right here, these drawings. And you can also put the lines here, and then you can label saying hydrogen bonds, right? So these are basically the points. Now, I'm going to re-emphasize the idea of the word. Whenever you see the word describe, drawing is allowed. And many times when you see the word describe, you will see in the bottom of the mark scheme saying allow diagrams. So whenever you see the command word describe, if you can't express in words, make sure to draw and annotate. You can get points with that. And this is pretty much it for the topic 2.4. Here are some examples of questions that I have not found in the IB previous exams, and short answers and long answers, but these can possibly come in the future. You can find the answer to these on the video lecture for 2.4. And this is it. Thank you for listening. 2.4 is done.